I'm Geoff Hamilton, CEO of Stemson Therapeutics.
I'm Cenk Suman, Chief Technology Officer at Stemson.
We're really excited to tell you about something I think is quite different than what you've heard from the rest of the folks here at the event, and that is our iPSC-based solution where we're capable of deriving the cell types that can generate hair follicles, and we're translating that into a solution that we hope to treat alopecia and hair loss for folks across the world. So I'm going to spend just a moment here telling you a little bit about the solution, because most folks don't think about hair loss as an indication in this industry typically. And let me see if I can advance the slide. There we go. So I usually don't need to spend a whole lot of time impressing upon folks the sheer scale of the hair loss challenge. And that is across genders, across ages, races, ethnicities. It's estimated about 60 million Americans experience hair loss. And that's over about 500 million people globally. And what a lot of us don't talk about is that for a portion of this population, this has a tremendous effect on them. And that is whether you're experiencing an autoimmune condition that is damaging your hair follicles, whether you're one of the unlucky few chemo patients who experiences a lack or a loss of hair from a permanent perspective, whether you experience scars or wounds or burns, or whether you're just someone who has male pattern or female pattern balding. The effect is tremendous on some of these folks, and it really impacts their mental health and their quality of life. And so much so that these folks take quite a bit of action on their own. They spend billions of dollars of their own money pursuing solutions to solve this problem. Some of those solutions that they pursue are an attempt to stop further hair loss. Some of those solutions try to excite the epithelial cells in your skin to reignite hair growth. Some folks even take as drastic of an action as going to a hair transplant surgeon, where you have quite an invasive procedure, scooping a hair follicle out from about 4 millimeters deep in the skin and relocating it to a new area of the head. But what all of these potential solutions have in common is none of them are actually capable of generating net new hairs. And that's what these people really need. And so, for the most part, these solutions do not solve the challenge that these folks want. And that's what Stemson hopes to bring, something brand new and really different through the market based on our IPS technology, is we would be capable with this solution of generating a de novo supply of hair follicles that is desperately needed in this market. And so now I'm going to actually allow Cenk to take you through a couple of slides that talks to you a little bit about the biology, the science, and the technology components that we're bringing together all into a single solution to go after this problem.
Thank you, Geoff. So I'm glad to have the opportunity to go over. Can everyone hear me okay? Great. Okay, thanks. So I'm excited to go over our technology to this expert audience. Thank you. There's some really fascinating biology here, and we've made some awesome progress, so I'll take you guys through that. So the hair follicle is an organ. It's a miniature organ with blood vessels, neurons, erector pili muscle, secretory system, and it's got some pretty incredible architecture. At the root of all that sits the dermal papilla, a tight cluster of cells derived from neural crest that controls the whole hair growth cycle and talks to the rest of the cells there. And in the bulge, you can see epithelial cells that can differentiate into keratinocytes and other cell types that help maintain function and hair growth. So for a number of reasons, this organ can cease to function, age, damage, sun damage, blood circulation. There's many causes. And the DP fails to signal properly, and the hair doesn't grow properly. There's actually one of the earliest publications from Colin Jehoda and his team was from 1984 in Nature, where they show that actual dermal papilla can grow hair. And I noticed that this is the same year that my PI at Stanford, when I was doing my PhD, Mark Davis, discovered the T-cell receptor that makes all the T-cell therapies I've been hearing about at this meeting possible. How come we haven't gone so far with dermal papilla? Our discovery was in 84. It's shown that it can make hair. Well, there's several reasons. So it's very difficult to isolate these cells, a very small, rare cell population. And it's very hard to expand them, and there hasn't been a way to really generate a scalable number of dermal papilla from iPSC until now. So the proprietary discovery that underlies Stemson comes from the ability primarily to generate DP, dermal papilla, from iPSC. And with the next slide, I'll go over some proof of concept in our early model, the mice, that shows you that the system works. So in nude mouse, in our early proof of concept work, we've shown that the cells function. In green, you can see keratin 14, which is a marker for the hair. And in red, you can see human markers. So we're mixing human and mouse cells here to show that iPSC-derived human DP can contribute to the hair growth. And for a number of reasons, mouse isn't the ideal model for this, so we'll talk more about that. And the hair is able to grow out of the skin, as you notice, because of the scaffold, biodegradable scaffold that we've engineered as well. I'll talk more about that. And the next slide will also take you through our process very briefly. So just to walk everyone through this, here's the concept. So we isolate primary cells from the patients. Here it's PBMC. It could be any appropriate cell type. And we plan to generate iPSCs from these by industry standard protocols like Sendai virus. And then we move these into our differentiation processes. We can differentiate DP from the iPSC. Also, we can differentiate epithelial stem cells. You can see the DP in red, epithelial stem cells in yellow. It's about a 3 to 4 week differentiation process, which is compatible with what we've seen in other models in industry, making medically useful cells from iPSC. And what we do after that is we load the organoids into the scaffold. Now, it's pretty amazing that these cells self-organize into organoids, and we're also developing in vitro assays to measure the function and potency of these organoids. And I won't talk too much about that, but if you're interested, you can come talk to us afterwards. And the proprietary biodegradable scaffold houses the organoid, and it has a rod or shaft component that comes out of the skin to allow the epithelial cells to grow downwards to heal that wound that's created through the surgery. And about the surgery, we're also working on ways to automate this. So we're exploring partnerships. I should let you guys know that there are already automated solutions available in the clinic, in the transplant clinics, to be able to do this. So we're working with these companies to be able to have an automated delivery solution as well. So as a CTO of Stemson, one of my mandates is to bring together different technology workflows to create better products for the patients. So I'll just mention a few of these very briefly. So we're doing a lot of stem cell engineering. We're working on our process. I'm happy to share that we signed a partnership with Dark Horse Consulting to do a development plan on our process. Second, we have a lot of activity with partners and in-house to engineer the biodegradable scaffolds to find the optimal design, materials, and the process so we can bring the best scaffolds to the patients. And this third one I'm really proud of. We're building a bioinformatics and data science team and hiring actively for that. So if you're doing that kind of stuff, please talk to me. But we're building ability, just to name two things that we're working on, ability to bring together genomics and imaging to be able to tell what the cells are doing. And second, to come up with good QC criteria so we can actually measure these before going into the product. And lastly, we're also working on robotic delivery solutions, as I mentioned earlier. And then with that, I'll turn it over to Geoff, who will take you through some of our clinical strategy, our pig model, which I didn't talk much about, and introduce our team.
Great. Thank you, Cenk. So as Cenk said, unfortunately for us, mouse models and mouse skin are just not appropriate to complete the development of this process. And what you see here on the slide is just a histological cross-section to kind of prove the point here that mouse skin in box D there is just simply too thin. It doesn't even offer a niche for us to really complete the parameters of the scaffold and some of the cell dosing range and testing that we're doing. So we're now actively testing in our pig model, which is what we expect to be our final preclinical model. And it's, many of the experts that have advised us on this sort of guided us in this direction, saying, pig skin is nearly identical anatomically and from a epidermis and dermis perspective to human skin. So we're really excited to be in this part of the journey to let us move as fast as we can towards the clinic and testing in humans. So with that pig model, we are initiating a roadmap of products. What's interesting about our indication is this is really a consumer-oriented market. This is a self-pay market, and as you think about that, we can conceive of many different versions of a product that would serve different sectors and customer segments, if you will. And so we do know that our fastest path to market, fastest path into clinical trial, least risky from a clinical outcome perspective is an autologous approach. And together with some of our industry partners, we've sort of gone out and really pressure tested that there's a strong market that would be willing to accept and pay for an autologous solution. And so that's sort of a first priority on the fast track, if you will. But we do understand, and the whole industry is working on allogeneic approaches, and as that matures, we expect that we'll spin up a program, hopefully in the not too distant future, to pursue an allogeneic program that would allow us to achieve some economics for a large base of customers that would want access to this solution. Now, one interesting thing is we are now sort of the leader in the space as it relates to making hair follicle cells, and that by itself as an asset serves as a platform that we plan to use in box number 3 here on this slide that opens up the opportunity for additional drug screening. And we know this is a large, diverse market that will likely have many different types of products that people will be interested in using at various stages of their journey with hair loss. And we don't expect that the appetite for molecules or drug-type solutions will go away. And so we are in a unique position to go about discovering additional enhanced molecules that might be able to be brought to market. And so a little bit of a shorter-term view in terms of how we're driving the company here is we're a Series A stage company. We're about 18 folks growing up to 25, hopefully, as we're trying to hire quickly. As we complete our work in the pigs and get to a higher state of confidence that we're ready for human trial, we'll then go out and look to raise the next fund to get us into human trial. And so that's a clear sort of focus for us. And some of the patients that we have our eyes on, really, as you look across the landscape of folks suffering from hair loss, they primarily fall into these 4 buckets. The big bucket being the one on the left, androgenic alopecia, that's male or female pattern balding. But we're also really looking into other causes of hair loss, whether that be from scarring, chemo-induced, that the hair never grows back, or some autoimmune conditions like alopecia areata. I will state that our solution is not fundamentally addressing an autoimmune condition, so that may be one of our more challenging buckets, but there are actually complementary drugs, such as a phase 3 Eli Lilly drug. It's a JAK inhibitor that if that comes to market and knocks down the autoimmune condition, it may provide an environment where we can come in and supplement any hair recovery that those patients need. Last slide here. I really just want to acknowledge the team. We have a very experienced and incredibly passionate group of folks that are working on the solution, and that includes board directors, scientific and medical advisory folks that are really excited about what we're doing. We're really lucky to have a great group of early-stage investors from both pharma industry with AbbVie and Allergan, as well as great biotech venture capital funds participating with us. And so we're looking forward to continuing on this journey, and I would just end off by reiterating something that Cenk said, that you can see sort of an interesting mix of different technologies and capabilities that need to intersect with this solution. And so we're always exploring various components, technologies, elements of the solution to incorporate into this rather complex solution. And so if anyone out there has ideas on technologies you may have or sets of knowledge you may have, I would just welcome you to come and talk to us. We're open to sort of expanding the realm of partnerships and relationships that we have. So with that, I thank you so much for your time and happy to take a question or two. I think we have 1 minute. Okay, great. Thank you.